I'd now like to introduce Dean Moscave. Moscave has served as Dean of the College of Science and Engineering since 2018 and has held various roles in the college for more than 45 years. Before serving as Dean, he was Associate Dean for Research and Planning in the college. And prior to that, he was the head of the Department of Electrical and Computer Engineering from 1990 through 2005. Dean Cave joined the Electrical and Computer Engineering faculty in 1975, and he has been a tremendous asset to our college ever since. He will end his term as Dean of the college in January 2022, and he looks forward to returning to the Department of Electrical and Computer Engineering. Dean Cave, you can feel free to unmute yourself and take it from here.
Well, thank you, Joelle, and thank you everybody for joining us. Good afternoon, and wonderful that you could be with us. This one, my last public lecture as Dean, and of course I look forward to attending the future ones, particularly with our new Dean, Andrew Allen, joining us in January. So it is really my honor to welcome you to this brand new public lecture series, Rising to the Moment. As you may have heard, the university successfully completed its $4 billion driven campaign this summer, and it has given us incredible upward momentum. As a college, the campaign has moved us forward in inspiring new ways, some of which you will hear in just about a few minutes. We have reached an impressive milestone, but the journey isn't over. It never is. We face the work of the future with the same optimism and determination that fueled us during the Driven campaign. We're more committed than ever to making undergraduate and graduate education an attainable dream for all students, reaching out to support our community, and advancing research to tackle the world's grand challenges. We will continue to rise. For the first lecture in this new series, I wanted to showcase a faculty member whose work truly represents rising to the moment. And as you will hear, Professor Chun Wang's research does just that. His lab has made incredible strides towards developing a vaccine of the future, an effort that began long before COVID-19 vaccines became a daily topic of conversation. But there is still work to be done to bring the promise of the research out of the laboratory into the world where it can have a broad and lasting impact. Today's science and engineering challenges demand creative and educated minds, innovators who ask critical questions and work tirelessly to discover the answers. As a College of Science and Engineering, we're uniquely positioned to address these challenges and opportunities head on. CSE's faculty and students are pushing forward the frontiers of discovery, redefining what's possible, and making transformational impacts on the lives and well-being of Minnesotans and people throughout the country and around the world every day. Today's speaker is just one example. And now I'd like to turn things over to Jane Lansing, a distinguished alumna and longtime member of the CSE Dean's Advisory Board, to share great news about the end of our recent fundraising campaign and to introduce today's featured speaker. Jane, thank you very much.
Yeah, thank you, Dean. And welcome to the alumni and students and friends of the College of Science and Engineering who are joining us today. I'm Jane Lansing. I'm a 1976 civil engineering graduate. I'm also a supporter of CSE, and I donate because my education led me to a career I never imagined. So I know that the engineers and scientists that we are educating today are going to contribute in ways that we cannot even dream of. As Dean Cave mentioned, the university recently concluded its 10-year Driven campaign. And it is my pleasure to announce that the College of Science and Engineering exceeded its $285 million fundraising goal. Over 12,000 people made gifts totaling more than $313 million. So thank you to everyone who generously contributed to the college during the campaign. All of these gifts add up to dramatically increased resources to support initiatives across the college, and particularly in three main impact areas. First of all, for students, for undergrads, doubling the number of endowed scholarships to 345 and increasing the annual scholarship funding by 138%. For graduate students, increasing not only the number of fellowships, but growing the base value, enabling a 71% increase in awards. Secondly, for faculty, creating 17 new endowed chairs and professorships, and increasing resources for faculty recruitment and retention by 27%. And then lastly, for facilities, renovation and construction of 5 research and teaching facilities, Shepherd Labs, Mechanical Engineering Labs, the Anderson Student Innovation Labs, the new Physics and Nanotechnology building, and the first floor of Lind Hall to create a CSE home. I saw a number of people signing in who graduated around when I went to school here. So if you're ever near the campus, you should stop in and see some of these fabulous new facilities. So thank you to all who contributed to the Driven campaign. Because of you, CSE is a leader in educating the next generation of scientists and engineers. So now it is my pleasure to introduce our featured speaker, Dr. Chun Wang. Dr. Wang received his PhD in bioengineering at the University of Utah, supported by an individual National Research Service Award from the National Institutes of Health. He conducted postdoctoral research at MIT before joining the faculty of the University of Minnesota in 2004. He's currently an associate professor and director of undergraduate studies in the Department of Biomedical Engineering. His research interest is polymer-based therapeutic biomaterials with applications in controlled drug delivery, immunotherapy, medical devices, and regenerative medicine. He has published over 100 peer-reviewed articles and reviews, and he has given over 120 invited talks. He served for 10 years on the editorial board of the Journal of Controlled Release and is currently an editorial board member of Advanced Drug Delivery Reviews, Pharmaceuticals, and an associate editor for Delivery Systems and Controlled Release, a section within Frontiers in Biomaterial Science. So please join me in welcoming Dr. Wang.
Professor Wang, I think you need to unmute.
Yes. Can anyone hear me? Can
That's
everyone hear
good.
me?
Thank
Yeah, am
you.
I audible? Okay, great. Thank you so much. Thank you to everyone for tuning in. I am extremely delighted and honored to be here to share our research with you. I'd like to thank Dean Cave for this wonderful opportunity, and to Jane for the very kind introduction and for moderating the Q&A. My title of the talk is shown on the slide. My lab is in the Department of Biomedical Engineering, a relatively young department in the college. Now, there. Earlier this year, we published a paper in the Journal of Controlled Release. The journal is a specialty journal that publishes research in the area of drug delivery. The title is filled with jargons. It says mucoadhesive wafers composed of binary polymer blends for sublingual delivery and preservation of protein vaccines. Now, we were at the time and still are in the midst of the pandemic, and vaccine was on everyone's mind. So we attracted some media attention, and this is what KSTP Channel 5 said about our work. Could be a game changer in how vaccines are administered. CARE 11 talked about us. They said the vaccine wafers placed under your tongue could help deliver vaccines rather than shots. And this video is still on YouTube for you guys to watch. Now here, I'd like to take this opportunity to share with you the complete story behind this work. And I would like to start with a brief background introduction on what polymers are, for those of you in the audience who may not be very familiar with the concept. And then I'll discuss some of the challenges in the delivery of vaccines and our solutions and results up to date. And then I'll share with you some of my thoughts about the future. Well, what is polymer? A polymer is a macromolecule. It's a gigantic molecule. The small molecules that many of us are very familiar with, such as water, H2O, carbon dioxide, CO2, if these are represented by these colorful beads, then polymers are strings of those beads that are made of small molecules that we call monomers. These strings of beads can be constructed in ways, as you can certainly imagine, of great variety and color and shape and size. And they don't even have to be all linear. They could have branches and so on. So this idea of polymers makes it possible to construct a vast range of materials with almost unlimited degree of functionality and structure, and could have important applications in many areas. This is an example of what these polymers actually look like. Notice that these are very wiggly worms. They are very, very small. The width of these polymer chains is on the order of millions of a human hair, very, very small. And the other thing about them is that they like to fold. They wiggle around and fold in 3 dimensions. And the 3-dimensional structure of these polymers can be very, very critical to their activity or function. Now, polymers obviously have a tremendous impact on our lives and on our economy. The plastics industry is a multi-billion-dollar component of our economy. And importantly, polymers are also the stuff that make up life, right? Living organisms, proteins, DNA, RNA, and polysaccharides. These are the structural elements of cells and machinery of life, genetic materials. These biopolymers are all, yes, like I said, these are actually also polymers. In a sense, a protein is no different, is very similar to a nylon from the polymer's point of view. So let's keep that in mind. Now, my lab at the University of Minnesota in the Department of Biomedical Engineering is focused on developing polymer-based biomaterials to solve problems in healthcare. We have a strong focus on using polymers to help improve the delivery of drugs. We're working on a range of projects. Some are related to cancer therapy, some are related to vaccines and infections. In the interest of time, I'd like to focus today's talk on our vaccine project, and perhaps we'll get a chance to talk about my other work in the Q&A. But if you'd like to know more about our research, please visit our website at biomaterials.umn.edu. Well, so about 6 years ago, I became acquainted with Dr. Mike Berry, who's the head of the Vector Engineering Lab at the Mayo Clinic. And through Mike, I got to know Dr. Jagan Sastry, who's an immunologist at MD Anderson Cancer Center in Houston. Now, Mike and Jagan at the time were interested in developing a vaccine for HIV. The idea was to take a protein, a glycoprotein called GP140, which is located on the outside of an HIV particle, and use it as a vaccine to produce anti-HIV immune response in humans. Now, at the very beginning, when we were exploring this idea, we immediately noticed two problems. One of them is that GP140 is administered to an animal, for example, by injection using a needle and a syringe. Now, this is usually fine. And in fact, HIV does transmit through needle injections. However, it's not ideal. The natural pathway of HIV infection is not through a needle injection, but rather through mucosal tissues, right? Through contact and breaching of mucosal membrane. Now, and it turns out that if we give the HIV protein by injection, it doesn't generate the type of immune response that is really necessary to fight the infection. So we thought, wouldn't it be nice to generate mucosal response by delivering the protein through the mucosal tissue? Now, the second problem is that GP140 is very unstable, whether as a solid powder form or after dissolving in water. Now, it's said that this protein must be stored at -20°C, and it's only stable for about 6 months, and must be kept at 4 degrees at all time. After it's dissolved in water, it has to be used immediately, and very soon it will lose activity. So that was obviously a problem. And so the delivery problem and the lack of stability problem of HIV GP140, they go way beyond HIV and this particular protein. Now, what about needle injection? I'm sure everyone can relate to this. It's painful, not pleasant at all. And in terms of vaccines, if we're getting booster shots and we have to come back and get another one, and that's not pleasant either. And in some cases, we may get a third booster, and so that triples the unpleasantness. Used syringes and needles are often disposed of, and they unfortunately could cause injury, and they pollute the environment. So these are just some additional problems. And of course, the use of these blunt instruments add to the cost of vaccines. Well, so unfortunately, the majority of the vaccines given to billions of people today are primarily still given by injection by needle and syringe. Here, I'm highlighting some of the very common vaccines that are given by injections today. Well, the stability problem. Again, this goes beyond just one particular protein. If we look at the many different types of vaccines currently being used or being researched, they're more or less all made of proteins, nucleic acids, and lipids, and these biomolecules are very prone to what's called denaturation. That is, when they lose their particular three-dimensional structure, they lose the activity. Now, this could happen under many situations, with slight heating, freezing. When ice crystals form, they can destroy the biopolymers, and by dehydration or by having too much water that can break down these biopolymers. So a very big problem, very prevalent one for all vaccine types. As a result of that, the standard way of transporting and maintaining vaccines these days, it relies on something called the cold chain. Now, this cold chain is an unbroken chain of refrigeration, starting from the manufacturing location to storage facilities, and all the way en route to hospitals and clinics, and eventually to the hands of healthcare providers. So the cold chain, right, the cost associated to that is estimated to account for about 80% of the cost of all vaccines. So this is a very significant component of the cost of vaccines. So the challenge that we were facing at the time when our interdisciplinary team came together was to see if we could come up with a simple polymer-based biomaterial that could do two things. One is to deliver protein vaccines, such as HIV GP140, without having to use needle. And number two, can we create this material to stabilize proteins like GP140 without using cold chains? And I emphasize simple polymer platform because we believe that simple materials will have a better chance of making impact in the world, going through manufacturing and scaling up, eventually going to the real world. Well, first, the solution to needle injection. We were drawn to a way of delivering drugs called sublingual delivery. So the sublingual site is the oral mucosal tissue under our tongue. Now, in particular for vaccine delivery through the sublingual site, it is particularly advantageous because the sublingual site is connected anatomically to a large network of lymphatics. So this network contains many types of immune cells. It's part of our immune system. So vaccines going through the sublingual site will have easy access to our immune system, and this is very good. Now, delivering drugs through the sublingual tissue is not new. There are currently many drugs on the market that are delivered this way, for example, nitroglycerin. However, the challenge here is that if we're delivering a gigantic protein like GP140 through the sublingual site, then these large molecules, being polymers themselves, are gigantic and sluggish, and they don't really cross the sublingual tissue membrane very well. So primarily for this reason, currently there is no sublingual vaccine approved and available on the market. And so we thought if we could maintain the presence of a protein vaccine on the surface of the sublingual tissue long enough to buy time for the protein to slowly migrate through the tissue membrane, we may have a shot. So if we look closer to the anatomy of the sublingual mucosal tissue, this is a cross-section of a tissue illustration of the tissue. Near the very top is layers of so-called epithelial cells. On the very top of the epithelial cell layer is a layer of mucus. If we look closely at the mucosal tissue surface and the mucus itself, we notice that the mucus consists of a type of protein called mucin. Mucins, because they're proteins, they're also polymers, and they're represented by these wiggly chains covering the mucosal surface. This is very important for us because it protects the mucosal surface. It gets rid of harmful substances and so on. It's turned over frequently, but this is also a very significant barrier if we were to deliver drugs through the sublingual mucosa. Now, our potential solution to this problem is to create a material that would interface with the mucin layer in a way that is analogous to Velcro. Imagine the mucin-covered sublingual mucosa is the hairy, softer, hairy part of the side of the Velcro. We would like to see if we can design or select a polymer that would work as the other side of the Velcro that would form an interface with the mucin. And we selected a polymer called carboxymethyl cellulose. Okay, this is the chemical structure of this polymer. And when we apply this polymer to the surface, the molecular chains of carboxymethyl cellulose, or CMC in short, will be able to interact with the mucin molecules favorably to form molecular bonding, so they're bound together. And they may also entangle with mucin just by wrapping themselves around, just like how Velcro works. So this tight interface or mucoadhesion should be able to allow the CMC layer and the vaccine that's included in the CMC to have enough time to diffuse across the mucosal surface. Now, I need to emphasize that the reason why CMC is so good at that, as I'll show you in a bit, is partially because that this particular polymer in 3 dimensions forms crystalline regions within itself. So it's a semi-crystalline polymer. These crystalline ordered regions where chains pack orderly brings rigidity to the molecule so that they're like the hooks of a Velcro that can stick into the mucin layer and form this nice adhesive interface. Now, the other problem of lack of stability of proteins, typically a protein is surrounded by a shell of water. And the shell of water is very important to maintain the stability and activity of the protein, just like we ourselves need to be hydrated, so our skin will glow and we will feel very comfortable. This is the same as all proteins. Now, when we heat the protein up or dehydrate it or freeze it and then dry it, if the protein loses the shell of water, it could alter its 3-dimensional structure and lose its activity. So our solution to this problem is by using a polymer called alginate. This is, again, the chemical structure of it. Alginate is a very common, safe biopolymer. It's used in food products. And the nice thing about alginate is that when alginate is applied to a protein, the molecular chains of alginate will be able to wrap themselves around the protein molecule, replacing the shell of water and forming a shell of alginate. Now, this protective shell helps keep the protein comfortable, maintaining its native 3-dimensional structure and hence its activity. And it's important to notice that the reason why alginate molecules can do this has something to do with the fact that alginate chains, instead of forming crystalline domains, these chains are completely structureless or amorphous, so that they're very flexible. They are free to move around in space and wrap themselves around to conform the surface of a protein molecule. Now, we spent quite a bit of time to play with these 2 key ingredients, and we realized that it's really important to strike the right balance between these 2 very important ingredients. Now, it turns out that CMC, while it's very good at providing strong adhesion with the mucosal surface layer, it is a terrible polymer to be in contact with proteins. It actually destroys native protein structure. So that's not good, not entirely good. On the other hand, alginate, although it's very good to form a protective layer for proteins, it is not very adhesive to mucosal membrane at all. So we experimented for some time, and we found that, yes, we need both ingredients and we need them in a balanced way so we can reap the benefits of both of them. So we designed a fairly simple and potentially scalable method to produce these polymer wafers using various ingredients, as I described. This is a picture of what these wafers look like. If we look closely at these wafers using electron microscopy, we notice that the wafer has many pores. These pores are partially by design, and it's inevitable because of the manufacturing process. We form these wafers using lyophilization, so these pores are left behind by water, ice crystals rather. And then the other potential benefit of having these pores is that when they're applied to a moist mucosal surface, the pores would suck the water up and form an even tighter interface between the wafer itself and the underlying mucosal tissue. So there's very good reasons why these wafers are made to have these pores. Now, the first question we ask ourselves is that, can these wafers actually deliver a protein to the sublingual tissue? Well, the answer is yes. Now, to test this, to answer this question, we set up an experiment where we used a protein, a model protein, bovine serum albumin, and we labeled it so that it's fluorescent and can be easily seen and detected. Now, we put the labeled BSA into our wafers and compared and adhered it onto a mucosal tissue. And then in comparison, we applied the protein in water, free water. We put a drop on the sublingual tissue as well. And then we compared how much the protein gets into the mucosal tissue. And as you can see, on the right-hand side in particular, this very thick and bright yellow layer is the BSA. That means the protein has crossed the mucosal, sublingual mucosal barrier and has gotten inside the tissue. On the other hand, the free protein did not do that. I should also mention that this experiment was done even after we flush the sublingual mucosal site with large amounts of water to simulate someone drinking or eating, and we saw the same response. There's a significant difference between the wafer and without the wafer. The wafer is significantly improving the delivery efficiency of this protein into the sublingual site. The second question is, well, can these wafers protect proteins against freezing, drying, and heat? The answer again is yes. So to set up this experiment, we took a protein, an enzyme called beta-galactosidase. This enzyme, when it's fully active, it can catalyze the reaction of a small molecule and create a color of dark blue. So this gives us a very nice way of detecting and quantifying the activity of this protein. So we created wafers containing beta-galactosidase and exposed it to various kinds of harsh treatment and compare the activity of the enzyme to another sample without the wafer, just the protein itself. So first, after freezing and dehydration, we measured the beta-gal activity. Without the wafer, 25% of the activity of the enzyme was lost. With the wafer, 100% was retained. We then freeze these samples and dehydrated them, and then we exposed them to 75 degrees C for 30 minutes. It's a pretty harsh heat challenge. Now, for the free protein with no wafer protection, 60% of the activity of enzyme was lost. With wafer protection, only 25% was lost. So clearly, the wafer protection, the wafers show a significant advantage than just a free protein in terms of protecting the protein against these harsh environmental treatment. The last and perhaps the most important question is, can a vaccine formulated in these wafers can actually be distributed without using cold chain? And can these wafers actually produce useful immune responses in the body? Well, the answer again is yes. So what we did, my students and I, what we did was that we took the HIV GP140 protein, 5 micrograms, and we encapsulated it into these wafers. And then we packaged them without any freezing or at other low temperature refrigeration. And we shipped them through FedEx from our campus here in Minneapolis all the way to Houston, Texas, to the lab of our collaborator, Dr. Sastry at MD Anderson Cancer Center. And after they received the package, they let the samples sit on the bench for 5 days at under ambient environment with no refrigeration. And then they apply these wafers to mice in the lab, and then look to see if the vaccine has produced any antigen-specific immune responses. And again, also by comparison, they did the same experiment to these mice using the GP140 protein that is freshly prepared, freshly taken out of the freezer and freshly prepared. So what they found was that in terms of the various immune responses, the parameters of immune responses, including interferon gamma production and various kinds of antibody production levels, the wafer formulated GP140 has equal or sometimes better immune response than the GP140 protein freshly prepared from a freezer. So this was very encouraging. We were very happy to this results. So this really demonstrates that these wafers do work. So what's next? One idea we're thinking about is that because vaccines today come in a great varieties, we have demonstrated, as I've shown you, that this wafer technology works with subunit vaccines based on proteins. We're curious whether the similar or this type of technology could be applied to nucleic acid-based vaccines, such as DNA vaccines or RNA vaccines. As many of you know, the first two COVID vaccines are based on messenger RNA. So these are mRNA vaccines. There are other kinds of vaccines based on viruses or live and attenuated pathogens. And these are all, they pose various kinds of challenges. So can we customize our polymer wafers so that they will also work with these other different kinds of vaccines? We have done preliminary studies on DNA molecules, which we hope to be published soon. And so if after customization we could work, then it would be a tremendous benefit to this technology. Now, another focus at this point is that we would like to see if this technology can be translated into clinical settings and make immediate impact on diseases that are currently already have vaccines for. However, these vaccines may be injected, they may have stability issues and therefore require the use of cold chain. And so if our wafers can be used to improve these existing vaccines for existing diseases, this may have more immediate impact to the world. Now, what about the future? We all understand the likelihood in the future of certain emerging diseases that are infectious globally. Now, it's estimated that these emergent diseases may arise more likely from particular parts of the world, primarily Africa, Central America, and South America, and parts of Asia. Now, unfortunately, if we ask how are we prepared to address these emergent threats, health threats, it turns out that these same regions and of course other places are really not fully prepared for such emergent diseases. So it is particularly important for us in our minds that we share this vision with you so that we can work together to address threats in the future. Now, our vision is summarized here. So should an emerging disease arises, then we hope that this disease can be quickly understood, the source and the pathogen quickly identified, and a vaccine can be developed. Now, after that, we will hope to be able to take the vaccine and formulate them into these wafers, and which will be able to be transported, disseminated globally without having to use cold chain at fairly low cost as a result, and eventually to the hands of healthcare providers. And the administration of these vaccines will be a simple sublingual administration without having to use needles and syringes. So this, in our opinion, could have tremendous impact on parts of the world that are lacking of essential resources, and even certain areas in the United States that are low on resources. And so to summarize, or to yeah, to summarize, this work is not possible without the dedication or hard work of my students and my collaborators. Sam Hansen, who got his PhD with me, grew up here in Minnesota in Eden Prairie, went to Northwestern for undergrad, and then came here with me to getting his PhD. And now he's working for Boston Scientific as an R&D engineer. So Sam has done the majority of experimental work in my lab that I've shown you today. And he's just a tremendous scientist and engineer. And Anthony Tabbitt was an undergraduate student working with me. He grew up here in Minnesota as well. And he majored in chemical engineering, but he became very interested in biomedicine. So he spent 2 months with me in my lab working on the wafer project, as well as a number of other projects. And he's now pursuing his PhD at MIT. And of course, I'd like to thank my wonderful collaborators, Shale, Jagan, and Mike, for all the great ideas, inspiration, and for doing the animal experiment. The project received funding from the Minnesota Partnership for Biotech and Medical Genomics, which funds collaborative research between the U and the Mayo Clinic. And I really thank you all for taking the time to tune in and hear my talk, and I look forward to your questions. Thank you.
Thank you so much, Professor Wang, for sharing that amazing glimpse inside of your research lab with us today. It's so inspiring to hear about the amazing advances that you're making. I'd now like to invite Dean's Advisory Board member Jane Lansing back onto the screen to pose questions to Professor Wang. If you haven't already, you can put any questions you have in the Q&A, and we'll get to as many as we can before 1 o'clock. So with that, Jane, I'll turn it over to you.
Yeah, and there's a lot of questions. So if we don't get to yours, I'm going to turn them over to Joelle and Professor Wang, and hopefully they can answer you guys personally. The first question is around how long this has to stay sublingually to ensure that it's absorbed. And I'm going to add on to that. Does it have a taste in the mouth as well?
Yes, very important questions. The first one, we wanted to develop this wafer technology to suit different needs. They can be, depending on the composition of the wafers, they can stick to the tongue for a matter of minutes before they dissolve, or a matter of hours. Now, with regard to the proteins, it took the wafers that we tested were able to dissolve after 1 to 2 hours. Okay, and that obviously was enough, as we've shown, for these proteins to enter across the sublingual barrier sufficiently to generate immune response. But that's another question, that is scientifically interesting. We would like to know, what really does it take? What is the maximum or the minimum retention time for protein vaccines to work? The second question with regard to taste, well, at this point we didn't put any flavoring into the wafers, but we certainly can. It's actually a common practice in sublingual tablet formulation these days. If you like orange, we could put orange flavor in there, or mint. It's totally doable.
Okay, another question. You looked at 2 polymers. Is there any need to design more polymers, or do you think that this is a fit-all combination of polymers?
Great question. We came up with 2 because those 2 have distinct purposes, right, as I described. My philosophy is to create a system that is as simple as possible, because my goal is to facilitate manufacturing, scaling up, and distribution, and potentially translation. Now, there's a need for a 3rd or 4th ingredient, that definitely is certainly possible. Especially since I mentioned at the end, there are so many different kinds of vaccines. Some are based on nucleic acids, some are based on the entire viral particles. And so we may have to, out of necessity, to add additional ingredients, or maybe changing to totally different polymer composition for those other vaccines to work.
Have you filed for patent protection? Is there a possible spin out startup company being formed to commercialize this?
Right. The initial two component wafers, we did not file patent protection because at the time we were very curious to see whether this idea would work, and we were using a model vaccine, and it just wasn't on our mind at the time. However, when we move on to a particular specific vaccine that has real impact in the commercial sector, for example, then it would be obviously advantageous to file for IP protection. We do currently have a startup company formed based on some of the technologies we developed in my lab, and this is part of the repertoire.
Okay, thank you. Have you interfaced with care providers to hear their thoughts on patient compliance with the sublingual approach and effectiveness in the face on non-compliance disturbance versus an injection administered by a healthcare professional? And I'm going to actually add on to that too. I think it's an interesting question. What about children? Does this work for children?
Yes, with the question about children, yes, it should in principle work for everyone. The sublingual tissue of children is somewhat different than adults. They tend to be more permeable, and so it might be actually even easier to get molecules across. And it's particularly appealing to children, and of course to parents, that we can avoid needle pricks altogether. And so this is tremendous benefit. I've personally talked to clinicians and healthcare providers, and they all unanimously agree that if we can get rid of the hypodermic needle, which is an invention that was around for many years already, it would be a tremendous benefit to a lot of people. I talked to, for example, the doctor who gave me the COVID vaccine about this. As I was wincing and holding my arm, getting jabbed with a needle, I talked to her and she's like, yeah, definitely, that would be a great idea. So.
But what about the compliance issue? Because I mean, if you could administer it, then the patient could walk out and remove it. Could they not?
Sure, in principle, yeah. I mean, I need to emphasize that any technology, as my personal opinion, any technology like this will have to take into account of patient compliance. Obviously, for life-saving medical interventions, we would like to reduce the barrier of patient compliance. But ultimately, if patients are not going to receive, are not willing to receive a medication or vaccine like this, it's not our job to force them to, and the purpose of this technology is to make it easier and more effective and less costly of a life-saving medication or vaccine, so that they can be accessible to more people in the world, and that's our goal.
And is the tablet uncomfortable, or can you feel it?
They shouldn't. Because these are about 5 millimeters across and about 0.5 millimeter in thickness, so they're pretty tiny. And after they are applied to the moist oral mucosa, it gets hydrated, and essentially it turns into a gel at some point. So it should be all right. There are similar sublingual tablets that are out there these days, delivering nitroglycerin, for example. There are these small molecule drugs. And so this is a fairly familiar form of pharmaceutical formulation that should be very well received by the industry and patients alike.
And how does the fluctuating pH of saliva impact the efficacy of the vaccine delivery?
That's a very important question. Fortunately, the saliva is not nearly as harsh as the content in our stomach or in the intestines, and so it's usually mild. In particular, it doesn't contain any proteases, so enzymes that are meant to degrade proteins. So proteins are in generally quite safe if they're in the oral cavity. Now, of course, patients may have, or people may have various kinds of oral conditions at the time of application. Some may be eating, some may be drinking, some may be smoking, yeah, so these are all important questions that we will have to consider when we really move this technology to an actual application.
Okay, and how does this relate to cancer treatment?
Yes. Now, in terms of sublingual delivery, let's say, if we're delivering a therapeutic, say an anti-cancer drug through the sublingual site, then the logical target would be something like head and neck cancer, where the tumor is located not too far from the sublingual site. So we can deliver chemotherapy, for example, to these tumors. Now, in the case of more commonly other forms of cancer, where they're remote from the sublingual site, then we're looking at systemic responses that are particularly important for certain therapeutic molecules based on the immune system. So for example, in my lab, we also work on cancer vaccines. So these are vaccines not to prevent cancer from happening, but rather to treat cancer. So they're therapeutic vaccines. So in that case, what we want to do is to generate systemic immune responses that our whole body is mobilized to recognize that a certain solid tumor is foreign and therefore mount an immune attack to it. So in that case, if we deliver that kind of vaccine through the sublingual site, because for its convenience and all that, and the fact that we have a vast network of immune cells around the sublingual site, that will give us an edge in generating the systemic response to combat many forms of cancer. So that's a tremendously good idea.
Okay. Have you tested for biostability and biocompatibility?
Yep. Again, a very important question. The primary, well, not a primary, one of the most important criteria for us to select these two particular polymers, right, CMC and alginate, is that we had biosafety in mind. So these two biopolymers are very widely used in food industry and also are in FDA approved ingredients for pharmaceutical products. And they're very well tolerated. They either break down in the body or be excreted from the body. They're very safe. Of course, if we combine it with a real vaccine, then the active vaccine ingredient will have to be validated in terms of safety. But in terms of the wafer technology itself, we're very confident that the materials we use are very safe.
And what would be the expectation if the tablet were swallowed accidentally?
Yes. Well, again, because it's very safe, alginate and cellulose-based materials are routinely used in food products. So if you eat them, swallow them, no problem. It'll just be fine. But of course, it's not ideal because the vaccine wouldn't have a chance to go through. Instead, the vaccine will be destroyed in the stomach and in the GI tract. So that's why we like to avoid it as much as possible to have strong mucoadhesion so the vaccines can go through the sublingual mucosa rather than getting swallowed.
Okay, so let's take one more question and then we're going to have to close out. So we're going to change gears a little bit. And what are other research projects using polymers that your lab is working on?
Yes, I would love to talk more about that, but here's just a little teaser. I mentioned that we're working on the delivery of therapeutic vaccines for cancer. We have, again, collaborating with a group at the Mayo Clinic. These are oncologists, and we're working on delivering molecules that would teach the immune system to recognize a particular type of cancer and then be mobilized to combat that cancer. We are seeing very good preliminary results in animal models, and we're trying to move again into translation. The second thing that I like to mention is that these polymers, like I shown in these strings of beads, they're just tremendous because we can build essentially any type of functionality and structure into them. For example, we have recently developed a type of synthetic polymer, artificial polymer, that actually they themselves can have active anti-cancer activities. And some of the polymers will be able to find a tumor cell, cancer cell, and punch holes into the membrane of these cells and killing them, blowing them up essentially. And that's a very interesting observation. We think about plastics and polymers being inert materials, but we've created these polymers so that they actually behave more like drugs and have very potent activity. So that's something that we're very interested in pursuing and moving forward at this point. Again, if you'd like to know more, feel free to visit our website, biomaterials.umn.edu.
And we'll include a link to that website when we send the follow-up email that will have a link to the recording of this presentation to make it easy for folks. We'll also include a link to the YouTube video that Professor Wang mentioned during his talk about the news coverage of this research, if you'd like to watch that and learn more. So I'd like to thank you all once again for being here today, and a special thank you to Professor Wang and to Jane. There were so many wonderful questions and there was a lot to wade through. So thank you, Jane, for getting us through so many. And thank you again, Professor Wang, for making the time to share this amazing research with us. I'm going to just close us out with a couple of final announcements. So I already mentioned we'll be sending a link to the recording of this presentation. It usually takes us about a week to get the video cleaned up and up online. But once we send that around, please feel free to share it with anyone you think might find it interesting. I think this is really inspiring research, and we'd be happy for you to share it with anyone else you think would find it interesting as well. I'd also invite you to attend some additional events that are coming up in the college. We have another one coming up just this week on October 21st. It's the college's 3-minute thesis competition, during which graduate students will have 3 minutes and only 1 slide to explain their research. And a winner will be crowned, and that winner will go on to the university-wide 3-minute thesis competition in November. So if you'd like to learn more, you can go to z.umn.edu/CSE3MT2021, and I believe that was put in the chat to make it easy for you. And we also have another noontime webinar coming up on November 9th, and that one will be a special behind-the-scenes webinar featuring the Minnesota Robotics Institute. And you can learn more and register for that at z.umn.edu/MN Robotics Institute webinar. That should also be in the chat, and we'll include the link in the follow-up email that will have the recording of this presentation linked as well. And for a complete list of all the college events that are coming up, which is always a very robust list, you can visit csc.umn.edu/college/events. And with that, I'd like to thank you all for attending and wish you a wonderful afternoon. We hope to see you at an event sometime soon. Take care.